Thank you all for coming. I'm actually probably not going to use the mic as long as you guys in the back can hear me. Can everybody hear me back there? Way back there? Okay. Great. Thanks a lot. So there's no denying it. Humans can destroy natural systems. And while maybe not as deliberate as roundhouse kicking a tree, increased land use and development associated with increasing human populations can give rise to changes in the environment, such as increased temperatures and pollution. And these can have negative effects on organisms. And we refer to these as environmental stressors. What makes matters worse is that recent meta-analyses suggest that stressors in combination most often interact with each other. Meaning that if all we know, even if we know the individual effects of stressors in isolation, this information is often insufficient to predict stressor effects in combination. Now, this is important because it can confuse ecosystem managers and our ability to conserve valuable systems, particularly those in which we would expect multiple stressors to co-occur. In light of this, coral reefs are of particular concern because their peak socioeconomic value as providers of fisheries and coastal protection, but they're also subjected to a variety of anthropogenic stressors. And many of these stressors are driven by the coastal population boom. So as coasts have more and more people on them, we urbanize coastlines more, we cover naturally absorptive soils with concrete. We also have higher densities of nutrient wastes, which are often improperly disposed of. And these two processes can contribute to nutrient runoff. And it's pictured here. It's the deposition of terrestrial-based sediments and nutrients into coastal systems like coral reefs. And so we know this can be an issue because corals compete with algae, and algae are often nutrient limited in these systems. So if we add nutrients, we can stimulate the growth of algae and have negative effects on the corals. Sedimentation, on the other hand, likely affects both algae and corals because it can attenuate light, it could inhibit gas exchange, and it could prevent settlement. Of course, nutrients and sedimentation aren't the only two things. When you have more people, you have more mouths to feed, and you expect more fishing, both for subsistence and for just plain fun. And so we know that the fish that are targeted by these efforts are often functionally invaluable on the reef, like this herbivore, and so removing these fish can have indirect negative effects on the corals. So nutrients, sedimentation, and fishing could have potentially important interactive effects on the reef system, though we don't know. But humans aren't the only thing that stress coral reefs. That's right. Nature can be a very scary thing, and nature can affect coral reefs through natural disturbances such as hurricanes and cyclones that can demolish large areas of reef and sweep them out to sea. And on the biotic front, we have crown of thorn starfish outbreaks that can devour large expanses of reef, leaving only skeletons behind. So we know that reefs have been able to persist for millions of years in spite of natural disturbances such as these, and over time they're able to recover back to their original states. What we don't know is how potentially interactive anthropogenic effects may affect the recovery of these systems, and whether or not they'll respond and rebound like we hope they would. This led my colleagues and I to ask the question, how do anthropogenic stressors affect reefs following a disturbance, with the hypothesis that we kick reefs when they're down. Human activities hinder coral reef recovery. And so to address this, we conducted a field experiment on the island of Moorea in French Polynesia. And we were interested in the three factors I just described: sedimentation, nutrient enrichment, and fishing. So we conducted a fully crossed three-factor design, which yielded all combinations of these stressors. It yielded 8 treatments. We had 9 replicates, so 72 units in the field. And our responses of interest were meant to mimic scenarios you might expect following a disturbance. So we looked at the growth and survival of small coral fragments that you might expect to persist or emerge following a disturbance. And we looked at settlement to barren substrate, which we would expect to be freed up following a disturbance. And we maintained the experiment for 6 months. So our coral fragments consisted first of Acropora pulchra, this thin branching species on the left, and this knobbier species, Porites rus, on the right. We attached these to cinder blocks with cable ties in the field, and on these same cinder blocks we put 4 settlement tiles. This is a picture of the unit from above, the full experimental unit. And we randomly assigned each of these units to one of the 8 treatment combinations I described a moment ago. For the sediment manipulation, we wanted to choose a high rate, but we didn't want it to be outside of what's been seen, so we looked to the literature over the wide range of observed sedimentation rates, and we chose a rate of 212 milligrams per centimeter squared per day. And we manipulated this by collecting sediments from a nearby river outflow, sterilizing the sediment, and then we used this high-walled frame that one of our German collaborator engineered to fit right over the unit. So you could pour the sediments out and have a very controlled and uniform deposition of sediments onto the units. And you can see the sedimented unit on the right. The nutrient manipulation was a little more simple. We just packed PVC tubes with 100 grams of Osmocote slow-release garden fertilizer. These conveniently fit right into the gaps of the cinder block on the unit. And we replaced these monthly. In units that weren't enriched, we just had sham PVC tubes. And just to show that the enrichment worked, we have nitrate plus nitrite concentrations in micromoles per liter on the y-axis, so water column nutrient levels. Controls on the left, and nutrients on the right, and the colors indicate different sampling days of a subset of the units. And we did indeed see an elevation of nutrients like we'd hoped. So the last manipulation was for fishing, and to manipulate fishing, we simply used exclusion cages of 4-centimeter mesh to keep out the larger consumers. That brings us to the results. So first, for Acropora pulchra, this is one of those instances where you don't even need to show the data or the stats to describe the first effect, which is that these coral were completely devoured by corallivores in all of the uncaged units in the first week of the experiment. So we went from having these nice little nubs that we collected from the field to having this really sad stump in all of the uncaged units, while all the caged units, like this one picture on the right, had 100% survival, so that was nice. So our Acropora pulchra growth data consist of only the ones that survive, so it cut our sample in half. But you can see here what we started to get, so let me talk you through this. So we have Acropora pulchra change in mass on the y-axis, and then we have 4 of the treatments that were caged. So this same color scheme will be used for the rest of the talk. Blue indicates units that didn't get fertilizer, green indicates units that did get fertilizer, and where you see this sort of sediment granule pattern, those are the units that got sediment. And so the one significant effect we saw is that there was a positive effect of nutrients on the growth of Acropora pulchra. If we then turn to Parietes roost, we see a somewhat similar pattern in that there was a fair bit of mortality. So let me talk you through this one. It's a little more complex. We have all the treatments here, all 8 treatments. So the 4 from before that were the caged units. These are the ones on the right panel, the ones without consumers. Panel on the left are the ones that were uncaged and exposed to consumers, which is why we have the fish here. Again, we're looking at change in mass, this time Parietes roost on the y-axis. These numbers indicate how many colonies were included in this analysis, and to be included we had to have 100% survival. So you can see out of the 9 replicates, very few survived out of the whole group. And the only effect we see is the same as what we saw in Acropora pulchra, a positive effect of nutrients on the growth of Parietes roost. But we can actually look at partial mortality and see whether survival was affected by our treatments in this species. So here it's the same treatment designations as before, but we have Parietes roost survival in % live cover on the y-axis. And this is where things get interesting. So we see a significant 3-way interaction here. And let me talk you through the most obvious effect that I see. That is that there is a negative effect of sedimentation in the presence of consumers, but a positive effect of sedimentation in the absence of consumers. And it looks like this effect is weaker when nutrients are present. So I'm going to return to this in a second, but before I do that, let's turn to the settlement tile data and see if we can get some clues as to why we're seeing this pattern. So the settlement tiles, we looked at coral recruitment and algae, but we didn't see very much coral recruitment at all and no clear signal across treatments. Algae recruitment and accumulation, on the other hand, was quite variable. Here's a couple of final photos just showing how variable it was. Some of them were cake and algae, some of them weren't. So here's a plot of those data. Algal dry mass scraped off the tiles on the y-axis. Again, all the same treatment designations as before. And again, we see a significant 3-way interaction, but in this case, it's driven by the fact that nutrients have a positive effect on algal growth only in the absence of consumers and in the absence of sedimentation. Okay? So if we take this into account, we can think a little bit about why we're seeing this 3-way interaction and come up with some ideas that make ecological sense. So we see that sedimentation has a negative effect on coral, which may be enhanced by or possibly even dependent on the presence of corallivores. However, if we take sedimentation and corallivores out of the picture, and we also take herbivores out, so we take all the consumers out, algae are able to accumulate and have potentially negative effects on coral. If we then have treatments where sediment was added, algae could never have accumulated in the first place, so we can remove the algae. And sedimentation could have an indirect positive effect on coral by removing the algae, similar to what the herbivores do. In light of this, we can think about our algae growth data that we just saw, and we would only have expected nutrients to have a positive effect on algae then in the absence of herbivores and the absence of sedimentation, which is what our data show, bottom right there. This makes sense ecologically, but it comes with the assumption that we would see an effect of algae on coral survival in our data. So when we look at that, this is what we get. So Paredes roost survival, percent life cover on the y-axis, algal dry mass on the x-axis. Not a clear pattern. It looks like there could be a negative trend, but there's a whole lot of noise in the data, suggesting that it's not as simple as I just described. There's likely other things going on. One more point of contention is the nutrient effect on corals differ for Paredes roost. So we see a general positive effect of Paredes roost growth, but we see a general negative effect, with one exception, on Paredes roost survival, meaning nutrients seemed to decrease the likelihood of survival, but if you do survive as a coral, you grow more. This suggests there could be multiple pathways or nonlinearities associated with the nutrient effect on these corals. So in conclusion, when we return back to our question, do we kick reefs when they're down? The answer is a complex one. We saw complex interactions among the three prominent stressors we tested here, but there's still questions to be answered from the data set. One thing that seems to be clear is that effects differ across coral species, so with different combinations of stressors following a disturbance, we may expect shifts in reef community structure. In worst cases, it looks like some stressor combinations favor algae and inhibit coral, suggesting that when disturbances happen in certain anthropogenic stressor scenarios, we may expect systems to not recover, but instead shift to more algal-dominated regimes with associated losses in key ecosystem services. So the last thought I'll say is that this study suggests it's really important for us to consider the multiple interactions that anthropogenic stressors can have in structuring these systems we depend on, especially considering that we expect all of these stressors to only increase with time. With that, I'd like to thank some really important people and funding sources that allowed this to happen. And this was a really cool collaboration I got to do with the French researchers at CRIO, the French station, the French Polynesia, which was a real pleasure. So thanks to them, and thank you guys for coming, and I'll take any questions. We have time for one or two questions, I'm told.
How frequently was the sediment dumped on the units?
The question was, how frequently was a sediment dump? So it was applied multiple times a week to achieve that daily rate that I described, the 212 milligrams per centimeter squared per day. Yes? I'm sorry, could you ask it again? Couldn't hear you. Great question. We did not, but there's a long-term ecological research station there, and they have data on general sedimentation rates on the back reef habitat that we worked in. That's where that 12 milligrams per centimeter squared per day estimate came from, so we used that as sort of a baseline. So we actually added that to our treatment. So our treatment was actually 200 milligrams, and we just added the 12. That's where that number comes from.
Thank you.
Thanks a lot.
Our next